Good evening, everybody. Can you hear me okay in the back? Yeah? Thumbs up? Nice. Welcome. I would like to acknowledge that the University of San Diego is on the ancestral homeland of the Kumeyaay people, and we'd like to pay respect for the citizens of the Kumeyaay Nation, past and present, and their continuing relationship with their ancestral lands. I'm Jesse Mills from the Department of Ethnic Studies and the Chair of Collaborative Research at the Humanities Center. And it's a true joy to welcome you to the university this evening for our fantastic lecture by our very special invited guest, Dr. Boland. So the Knapp Chair of Liberal Arts is a program that was established by a very generous endowment by Mary and Churchill Knapp in the mid-1990s. And this endowment has allowed the college to invite prominent speakers and guests to teach classes, to interact with students, and to collaborate with our faculty, researchers, and students here at USD. And so we're so excited to have you joining us. The Illume Speaker Series is an initiative of the College of Arts and Sciences and features not only our outside prominent thought leaders and guests, but also our own faculty, and it participates to inspire lifelong learning in our surrounding communities. So it's a true joy, again, to welcome you to USD. And now, to introduce our speaker, I would like to welcome and introduce Ryan McCool, who is a biochemistry major here at USD. And please give a welcome to Mr. McCool.
All right, it is my great pleasure that I'm able to introduce Dr. Linda Boland, the University of San Diego's current Knapp Chair for Natural Sciences and Mathematics. Dr. Boland began her academic career at Lafayette College, where she received her bachelor's degree in biology. She went on to obtain her PhD in neurobiology from the University of North Carolina, Chapel Hill. She then completed 2 postdoctoral positions before performing 10 years of research at the University of Minnesota Medical School. Dr. Boland currently holds a position as an assistant professor at the University of Richmond, where she has served an instrumental role in developing and maintaining the Integrated Quantitative Sciences course, a highly successful first-year team-taught course that combines chemistry, biology, mathematics, physics, and computer science through interdisciplinary topics in a single course. This program has played a pivotal role in not only increasing the inquisitive nature of the University of Richmond scientists, but also encouraging first-generation and underrepresented minority students to further pursue STEM majors. Her dedication to the interdisciplinary science extends outside the classroom and into her research as she explores ion channels through the connection of neuroscience and molecular physiology. Due to her tremendous work in this field, Dr. Boland was invited to partake in research at the Genelia Research Campus of the Howard Hughes Medical Institute as a visiting scientist during the fall of 2018. Beyond the laboratory, Dr. Boland has also worked to address the intersection of science and the humanities, a passion displayed by her role as a co-coordinator for the Forum on Bioethics, Race, and Healthcare in the fall of 2017, and an inspiration for tonight's talk on neuroethics. Now, while the cold weather of the Northeast may have been a factor in her decision to spend some time at USD, it seems sunny San Diego has decided not to cooperate. To make up for the cold weather, I ask that you please give a warm welcome to Dr. Linda Boland.
Wow, thank you, Ryan. I'm going to turn this microphone on here. That's about the best introduction I've ever had. You can do that every time. Well, thank you, everyone. I'm really honored to be here visiting the campus as Knapp Chair. I've decided after a week of walking around campus that I'm going to put in a petition to call this the most beautiful campus in the U.S. Okay, that was my best joke. I know you've already won that award, and I think it meets up to that. So I'm honored to be here visiting and working with the faculty and the students, talking about interdisciplinary science curricula, doing a little bit of teaching in neuroscience, working with some students, talking about faculty development, talking about women in STEM and strategies for success. So we've kind of got a whole list of things that I can do while I'm here for a few weeks. So thank you for all of the effort that has gone into arranging all of this. And in particular, I do want to thank the Endowment Fund of the Knapp Chair of Liberal Arts. I want to thank the Humanities Center, Dr. Brian Clack, Jesse Mills, Lindy Villa, the College of Arts and Sciences, Dr. Noel Norton, Dean of the college, and the Research Working Group, which has a large number of faculty involved and some students, headed by Joseph Provost, Jessica Bell, Divya Sitaraman, and a bunch of other folks. I'm not going to list everybody here. I also want to thank my home institution for giving me the opportunity to be on sabbatical. That's a generous opportunity, and so it allows me to be here for part of the time during my sabbatical. I won't talk too much about my research today, but just a little bit, and that research was largely done by undergraduates in my lab, as listed here, and was initiated with some work from a collaborator at the University of Pacific and Virginia Commonwealth University, and my own research receives funding from the NIH, and some of the students have other funding, such as Beckman scholarships. So we are here today to talk about how science and the human condition intersect. Now, I'm sort of going beyond my research area a bit into something that intrigues me about how my understanding of neuroscience and electrical activity in the nervous system allows me to think about research and clinical approaches that affect the human condition. And my hope is that by doing this for a broad audience, that there's going to be a little bit of something for everyone in here. An opportunity for us to engage in some thoughtful discussion afterwards, to raise some questions, perhaps to raise more questions than we are able to answer, but that's okay, I think. In this process tonight, I hope that you will learn about two key principles of neuroscience. How did I boil neuroscience down into only two principles? I did it, just for tonight. How do these apply to the regulation of behavior? And I'll show some applications of that mostly from research of other people. And then raise some questions for thought and discussion about how these applications might concern us in terms of how they might affect the human condition in ways that we care about, whether we have unintentional consequences or decisions to make about human identity and authenticity. And it probably does not need too much introduction to say that I was intrigued to see that your series in the Humanities Center this semester on the idea of truth, a session that I went to last week, and I know there's another one tomorrow as well, was interesting to me because I think that some of what I have to say speaks to the idea of what do we consider to be our true identity and what do we consider to be truthful in terms of experiences that we have and that we report on throughout our life. Now, I do want to make a really important disclaimer here. I am not a physician. I have never trained as a physician. I've never trained in clinical practice. There are things that I will talk about today that are clinical applications, and there's always the fear that people will think that I'm making a medical recommendation, or perhaps they will take what I say as a medical recommendation. Please don't do that. There's no reason to do that. What I'm trying to do is make us aware of some of the things that are happening, and hopefully, if you're more interested, you will be interested to explore some more on your own or through interactions with other people who do have that sort of training. I do have an awful lot of references that I used throughout my thinking here to prepare the material, and I am willing to share the details of all of these references with anyone who wants more information. Okay? So let's go ahead and get started. I am talking today about the human brain. How are the lights? Are we doing okay with that? Does this help if I go down one notch, maybe get a little bit of the light off of it, because we've got to get a little more contrast. Is that okay for folks? Good. Is it okay for me? Okay. So what I want to talk about is the human brain. This 3-pound soft mass of tissue that is about 78% water, about 10% protein, about 10% fat, a little bit of carbohydrate, and a number of other molecules. It is the seed of our existence. It allows us to have all of the cognitive functions that we experience: thoughts, emotions, belief, imagination, memory, about just about any cognitive function that you can imagine. How does it differ from the brains that are shown here in other vertebrate species? I think you can immediately see that there's a difference in size. These are shown to scale, for a relative scale to each other. And you can see a big difference in size. And the other thing that you see is a very big difference in the surface organization of the human brain. These immense gyrations and convolutions that you see on the surface expand the surface area of the brain tremendously. Within this cortex, as we call it, this 3 millimeter outer layer of the brain, many, many more cells can be packed and many more connections can be made than in the smoother brains that you see up here. I'm not sure if anyone in the audience, or some people have, but maybe some have not seen a human brain. This is a model of a human brain, not a real brain, but it is a similar size to a human brain, and I'm going to pass this around so that you get a chance to see the general organization at a gross anatomy level and the general size, although it's about the right size for a human brain. Don't worry about the color coding. That's not significant for what I'm talking about today because the color coding doesn't match some of what I'm going to talk about. But I can pass this around if anyone wants to take a look at it. Thank you. So one of the things that I will be talking about, I'll start talking now about one of the key principles that I want to communicate today, all right? And to do that, we're going to look at an old map of the human brain produced in the early 1800s by a German scientist, Franz Gall, or actually he was a physician. This is a map of the adult human brain that is referred to as phrenology. And this occurred at a time when people thought the human brain was like a muscle, and it was like a muscle that could expand its size and shape as it got used more often, as muscle tissue will do. And what he reasoned was that the surface of the skull had little bumps on it, and each of those bumps represented a personality trait of an individual, and the size of the bump would change when the personality trait changed over the lifetime of the individual. And you can see how in this map here, you can see a large area for cautiousness and destructiveness and secretiveness. So this is an interesting individual, I think. This turns out to be pseudoscience. There's really no organization of the brain as shown here. Also, the bumps on the brain don't actually map to the contours of the gyrations of the surface of the brain. And it turns out that this is not an accurate map. But one thing that is pointed out in what is now seen as almost like a comical representation of the brain, one thing that's important here is that it's a little bit visionary to think about the idea that some of what makes us human is actually encoded within the brain and that it is somehow functionally organized. And that general understanding, that general idea is one that plays out in future research. A more interesting aspect of this when we look at the surface of the human brain was discovered by Paul Broca, who was a neurosurgeon who had a patient come to him with a type of aphasia, a language disorder in which the person could not speak fluently and coherently. Speech was broken up into little small parts of words, and speech was hard to manage, hard to create. This probably occurred after some kind of trauma, such as a stroke. We know this area of the brain now to be called Broca's area because upon post-mortem examination, when the patient died, Broca found that this particular area of the brain had a lot of trauma to it, and he assigned this an area that was involved in language production. Subsequent to that, one of my favorite neuroscientists in history, Brodmann, took sections of brain tissue from the cortex of a variety of different animals, all post-mortem, human to small animals like mice, and he sectioned these into very thin microscopic sections, stained them with dye, looked at them under the microscope, and started to notice that he could characterize these different sections based on where they came from in the brain tissue, where he had taken the piece of brain tissue from, and he saw different patterns of organization. Each of them had layers of cells, of nerve cells, and he saw differences in the size of the cells and the density of the packing of the cells and the types of layering that occurred. And in this, he characterized more than 50 areas of the brain based on this cellular organization and then laid them out into this plan here. When he first did it, there has been a small refinement of this over time, but for the most part, it has held up over a century of research. These areas are now referred to as Brodmann's areas, and you can see, for instance, this area here, 4445, is Broca's area, as numbered by Brodmann. So together, stories such as this allow us to see that the brain is functionally organized. We know that if there are areas that are damaged, that there will be specific changes in the behavior of the animal based on the area that is damaged. For instance, if this area is damaged over here, area 17, that happens to be primary visual cortex, and the person would be blinded by this. If an area is damaged over here in auditory cortex, the person would have difficulty with hearing. If an area is damaged over here, the person may have difficulty with movement because this is the movement area of the cortex. So we do have these general categories of functional localization. So the first key principle to remember is that the brain is functionally organized, and we'll make use of that principle as we go on. The next thing we want to understand is a little bit about how the cells within the nervous system communicate. Within the brain, that 3-pound mass that we talked about, there are 86 billion nerve cells, and we think approximately a similar number of supporting cells. These are often referred to as the glia of the nervous system. This is a general picture of a cell. They don't all look like this, okay, but this is a schematic of a cell where we have the cell body with the nucleus, and we have an organization here that almost looks like a tree with branches over here, roots over here, and a trunk connecting them. I'm not going to have time to go into all the details of cellular function, but I do want to convey the name of this particular area of the nerve cell called the axon, and it's the trunk. And this is the area in which we know that there is electrical activity. There can be electrical activity throughout the nerve, but the generation and the movement of this electrical activity along this axon is critical to the function of the nerve cell. It's similar to the movement of electrical activity along a power cord that you stick into the wall and then plug into your laptop. And that charge that moves through there is similar to what is happening in an axon. How do we know this? Well, one reason we know this is because of some of the seminal work that was done using this fantastic organism called the squid. This is an Atlantic squid that was studied out in Woods Hole in Massachusetts. You have squid out here in the Pacific Ocean as well. Either one of them make good calamari. But when you eat it now, hopefully you'll remember something else about the squid, and that is that it was the source of tissue for studies that identified the nature of the electrical activity of the nervous system. And this is the nature of the electrical activity. This transient, brief change in voltage across the membrane surface of nerve cells. We refer to this as an action potential. Sometimes people call it a spike. It looks like a little spike of activity. Here you can see the size of the activity, the amplitude in terms of voltage. 1/10 of a volt is the total amount of charge that moves. That is similar to about 7% of a AA battery. And this occurs in fractions of a second, 2/1000th of a second or so 2 milliseconds, okay? This represents the activity, the electrical activity of that axon running all the way down the mantle of this particular organism. And the axon in the squid is so large, referred to as the giant axon. This is not a giant squid, that's a different organism, but the giant axon, when dissected from this, could be studied and be found to have this particular electrical activity that would be generated and regenerated over time. And that is the nature of the signal that we're talking about, sort of the currency of the nervous system, the action potential. Now, it turns out that you don't have to just use a squid, because this action potential is the universal currency of the nervous system and used in muscle as well. And it is found throughout every organism that's been studied. And these are the ones that are studied most often in research labs. The ones from human would be in tissue studied during neurosurgery, so it's a little different. In many of these are studied in animals that are used in experimental procedures following the proper controls for care and use of the animals and certification of that for ethical use of animals in research in any of these experiments. I do want to demonstrate to you that that action potential that I showed you can have different forms, okay? And this is really important for our story today too. So here are some nerve cells from 3 different regions of the brain. These are the recordings from nerve cells of 3 different regions of the brain. The red traces here show you the single action potential, similar to the squid one. And then on the right, in purple, you see series of action potentials, or sometimes no additional action potentials, or a series with a different frequency or time dependence to them. What this points out is that while that general currency of that large change in membrane potential, that large difference in voltage across the membrane, and while it is always generally very brief, there are differences in the shape of it, there are differences in what happens after the peak has been reached, there are differences in how many can be generated and how close together, and in the frequency. It turns out that this is due to the complexity of the different cell types in the nervous system and the types of membrane proteins that they have that make up this, that contribute to this change in voltage, okay? In the single squid axon, there were only 2 types of ion channels, and so the action potential always looked the same. But throughout the nervous system, what we find in different areas of the brain that are functionally organized, what we find is that cells in that particular area of the brain tend to have the same type of firing pattern, if we can call it that, this type of electrical activity. So the cells in this particular area of the brain, identified to be Purkinje cells in the cerebellum of the brain, they all have activity like this. Cells in the hippocampus, in this particular area of the hippocampus, all have activity like this, and cells in the basal ganglia have activity like this. So this is really important. Different nerve cells in different regions demonstrate different patterns of electrical activity. It's always electrical activity that's important here, but there are different patterns. In my own lab, we were interested in this question. I will show you just a little bit of data from my own lab, and I'm giving a research talk to the psychology students next week. So I'll have a chance to go into more detail about the work done in my lab. But I did want to introduce the concept that we're interested in this electrical activity and its complexity and how this electrical activity becomes so different in different cells. And so the way we approached this was to take a frog. This is a South African clawed frog that is a laboratory animal maintained in the lab, not shipped here from South Africa, although that's probably how we originally got them. They are maintained in the lab. And as you probably know, amphibians have external fertilization, and these egg cells of a female frog can be released into the environment. Here's a collection of egg cells shown with high magnification. They're about 1 millimeter in diameter. These are unfertilized frog egg cells. They are not nerve cells. They have no electrical activity. They do have the ability to generate electrical activity if we take the genes that encode for an ion channel, and in the laboratory, we take the DNA that is that represents the genetic code for that channel, and we prepare messenger RNA in a test tube. We inject this into the frog egg cell, and the frog egg cell will take that messenger RNA just as if it were a nerve cell. It will process it by translating the messenger RNA, assembling the different parts of the protein, trafficking it to the membrane, and inserting it into the membrane in a way that it can now conduct electrical activity across the membrane. However, until we did this study, nobody had ever seen an action potential in an egg cell. We would study the individual channel types and even mixtures of channel types, but we didn't have a sense that we could actually study an action potential until we did the experiment. And it turns out we can get an action potential in an egg cell. And this is our big aha moment. I mean, you can't imagine the exhilaration that we had in the lab that day when we first saw this, because really we didn't expect it to work. It's the kind of thing that we dreamed about doing, but we never really expected it to work. But we figured out what had to be done for this to work. And since then, we've been on a trek to try to figure out why do the action potentials look so different in different cell types? Now we have a method that we can employ to do this. We can't do this in the squid axon. It's too simple. There's only 2 types of ion channels. We can do this in the nervous system of animals, and people are doing that. But the way they do that typically is they take a complex nerve cell that has a complex, they take a cell that has a complex firing pattern, and they find ways to disrupt, inactivate, or destroy individual types of channels one at a time, and they take that away and they say, what does the complexity look like now? The loss of a particular phenotype in that electrical activity must be due to the thing that we took away. And that's not an unreasonable approach, but we have a different approach, and the different approach is more of an additive approach, as I call it. It's an approach whereby we take each of the components that might exist and we start mixing them back together, and we say, what does it look like now? And what does it look like when we add these different components in different ratios? And what's really interesting to me is not just that we can get an action potential, but lo and behold, we can get some really complex waveforms like these in an egg cell. This is published in this past fall in the Journal of General Physiology, and the recordings were done largely by undergraduate students in my lab who really enjoy doing these experiments because they're very exciting, as we call it. Okay, yeah, okay. So what I would like to convey is the second principle, that not only that the nervous system uses electrical activity, but I would be remiss not to call it an electrochemical organ. When we talk about one nerve cell and the action potential that is generated in its axon, that's its electrical activity, and it might generate one or multiple action potentials in a pattern, a cluster. But once you invoke a second nerve cell, what you have to ask is, well, how does this information of electrical activity convey to the next one, such that in these complex circuits, we can extend that information. We can have one nerve cell communicate with another. Well, it turns out that that communication requires chemicals as well. And so I've depicted that with these blue dots here, representing these chemical molecules that are released from these roots of the tree of this nerve cell. They are released into the space, a small space between these cells. They are received by a protein in the membrane of this cell, and then this cell, if the signal meets certain criteria, may generate its own action potential. Okay? And so each cell has to generate an action potential if it is to be involved in the circuitry. If this cell fails to generate an action potential, then the information doesn't flow along this pathway. So we could interrupt this communication by disrupting the ability of any one of the cells in a circuit to generate this action potential, or we could activate circuitry by getting any one of the cells, presumably one at the beginning of a circuit, let's say, to generate an action potential, which, if it was conveyed faithfully through this electrochemical conversion, electrical to chemical to electrical, then the signal would be transferred from one to the other, okay? So that's the outline for the second key principle, that the brain is an electrochemical organ made of these nerve cells that generate electrical activity, transfer that activity to chemical activity, and then back to electrical activity. Okay? So if these two principles hold, what I'm going to combine them to say now is that the electrical activity, which we've just talked about, in different functional regions of the brain, which we've also talked about, can be modified to alter the behavior of the organism. We know that these are the signals that are important. We know that they are generated differently in different areas, and if we can regulate those properties through those two principles, then we may be able to modify the output, and the output of the organism is its behavior. So here now I'm going to jump from some of the basic neuroscience information to some modern day uses of electrical activity to manipulate human behavior. And I'm really making a big jump because there's a long, sordid history, actually, of science and pseudoscience that has used electrical activity to try to regulate behavior. And some of it's a pretty horrific history, actually. I'm jumping, though, to modern day and only looking at really like the last 20 years or so, where significant advances in our ability to regulate behavior have occurred from the application of these principles, functional localization and manipulating the electrical activity of nerve cells. So here's an example from deep brain stimulation, which we can abbreviate DBS. These are magnetic resonance imaging pictures from the human brain. This is from a brain from the rear view. You see the outline of the brain here and some of the convoluted areas. And here is a side view of the right hemisphere of the brain, looking at it from the left side. What you see in these cases are outlines of where very thin wires serving as electrical conductors were placed into the brain during neurosurgery. And this was done in individuals who have developed a refractory type of Parkinson's disease that is no longer responsive to chemical treatment, which is one of the methods of treating Parkinson's disease. But after periods of time, and some people are never very successfully treated with this, but even those who are in the beginning, they often do not have a lasting recovery from the chemical treatments. So one technique that has been pioneered is to use deep brain stimulation to insert electrodes into the brain into areas that we know are functionally identified to involve the symptoms of Parkinson's disease, the motor behavior problems, and then to stimulate those areas of the tissue and see if you could relieve the symptoms. And this is where I actually got pretty interested in the clinical applications of electrical activity, because I saw a patient who had undergone this sort of treatment. And it was incredibly impressive. I mean, an individual who has the characteristic tremor at rest of Parkinson's disease, the rigidity of movement of the muscles, and the shuffling of the feet. Okay, after the neurosurgery and the implantation of these electrodes, which are then connected by a wire to a pacemaker-like device that is implanted underneath the skin in the chest, the physician can control the electrical activity of those electrodes through this device that is computer-controlled, and a patient can go from having the Parkinson's disease symptoms to nothing. Perfect movement. It's incredible. It doesn't necessarily work with everyone. I'm not necessarily making a medical recommendation here, but it does work in a number of cases. And it's an incredible application of electrical activity applied to specific brain regions. This has been mostly used in the study of movement disorders like Parkinson's, essential tremor, dystonia, Tourette syndrome, and it's even been used in some other disorders of behavior, such as behavior and control, such as epilepsy, intractable pain, severe chronic intractable pain, severe debilitating headache that are lasting and controlling of one's life. And in cases where this has been applied, there's been, in many cases, a number of success stories. And this tells us that these general principles of controlling the electrical activity and doing this in specific areas that we know are involved in particular functions of the brain can be successful. So the primary goal has been to rebalance the electrical activity of the nervous system through selective and reversible manipulation of electrical activity. Now, one of the current applications of deep brain stimulation is to apply it not just to movement disorders such as Parkinson's or severe seizure disorders and epilepsy, but to apply it to a whole host of mental health disorders, things like obsessive compulsive disorder or addiction, eating disorders, chronic depression, anxiety, post-traumatic stress disorder. So a number of different disorders are being looked at and have been used, actually, I should say, have been subject to deep brain stimulation. Let's look at an example here from the treatment of treatment-resistant clinical depression. And this work was first done in 2005. There were only 6 patients in the study, and 6 of them showed striking improvements in behavioral measures. This is pioneering work done by neurosurgeon Helen Mayberg, first at the University of Toronto, now at Emory, I think. And it involved neurostimulation using deep brain inserted electrodes into this area known as Area 25 on Brodmann's map, an area that has been sometimes dubbed now as the happiness center, because stimulating this with electrodes will make an individual who has depression, well, can make, I should say, not all patients will respond, but can make the individual experience a lifting of those severe symptoms of depression into a period of a neutral state, and perhaps even a period of intense happiness, and perhaps even a period of euphoria, depending upon the intensity and duration of the stimuli applied to area 25. The outcomes were so positive in this initial study that a number of other medical centers wanted to study this, and they designed a blinded clinical trial, because that's not what was done the first time. And in this trial, 200 patients were enrolled at 15 medical centers across the US. 2 years post-surgery, about 50% of them show 40% improvement. Some people have written this off as, well, that's not good enough when we look at harms and benefits. Some people have said, well, that's pretty amazing, considering that we're also talking about the people who are resistant to other treatments and have no other options, and are the worst cases possible, the worst cases. It hasn't really been tried on cases that can be addressed by other methods, such as drugs, medical intervention in that way, or behavioral modification, or combinations of those. I want to show you what a patient has, one example of what a patient says during this surgery. And if you're really interested in this, there are a number of different videos that you can find online, even videos showing the surgery in action. And interestingly, what I did not mention is that during this surgery, when these electrodes are being implanted, some of you may be familiar with this, but in fact, the patient is awake and alert during that surgery. It has to be that way because the neurosurgeon needs to know that those electrodes are getting into exactly the right area. It's pinpointed by various imaging techniques and by knowledge of where that area is. But of course, there's going to be some fine differences that represent human variability. In addition, there may be some variability in the surgeon's technique, and there may be other sources of variability. And in addition, the physician does not want to damage other areas of the brain that might be absolutely critical for certain functions, such as speech or hearing or something like that. And so the individuals are awake and alert and reporting out on what they are experiencing during this surgery. So it's pretty incredible. The other reason that this works well to do this is that the brain itself has no pain receptors. So once the anesthesia has been used to drill the hole in the skull, the small hole, to get the electrodes past the skull and the subcutaneous, the skin, the subcutaneous tissue, and the bones of the skull, once it's entering into the brain, the individuals don't experience any pain. So that's a good thing. So just to give you a sense of the one positive outcome of this and how impressive it was for one of the early patients that was studied, I'll read for you from a patient speaking during the placement of the electrodes in that area 25 to treat her devastating, clinically unresponsive depression. It was literally like a switch being turned on that had been held down for years. All of a sudden, they hit the spot, and I feel so calm and so peaceful. It was overwhelming to be able to process emotion on somebody's face. I had been numb to that for so long. What a difference it made in that person's life. Now, I want to raise an issue here now. And that issue, though, is that whether that deep brain stimulation raises any ethical questions about the manipulation of happiness. And this is with respect primarily to the deep brain stimulation being used to enhance happiness in patients who do not qualify for this based on clinical criteria. What might be our concerns about that? Do we have any concerns? Well, one question would be that there might not be a concern if we consider that voluntary, uncoerced, autonomous decision-making about changing one's mood and becoming happier can occur. On the one hand, we can hardly argue against this, and we do things already in our lives to try to improve our outlook on life and improve our happiness and improve our mood. We use mechanisms such as yoga and mindfulness training and exercise and vacations and all sorts of things. If the end goal is the same, could we rule out the possibility that someone could use deep brain stimulation to bring their affect in the positive direction, to have greater happiness? Would we care about the method that was used for this if that result is achieved? Part of the concern that we might have with this is that enhancing happiness when there is not a debilitating illness imposes a human will upon something that we might consider to be a natural state. We intervene when that natural state becomes really skewed, when it's too disturbed, when there is untreatable depression. And we have to ask if those conditions are, for an individual, are so outside the norm that they would be considered to be warranting this to relieve pain and suffering and to prevent further debilitation. But if an individual desires greater happiness and is not in that outside of that normal range, should it be allowed? And the question here is about what it means to be true to yourself, to oneself. In a similar way, we could ask about other types of manipulation of the brain, and I'll do that momentarily. I do want to introduce in the concept of this concern about using the tool to elevate happiness in individuals who may not need it. Comes up in Dr. Mayberg's quote here as well. In applying the technology, she says, "It's not my job as a neurologist to make people happy. I liberate my patients from pain and counteract the progress of disease. I pull them up out of a hole, and I bring them from -10 to 0. But from there, the responsibility is their own. They wake up to their own lives and to the question, who am I?" So with deep brain stimulation, we intervene when this natural state becomes very disturbed. Other conditions such as post-traumatic stress syndrome, stress disorder is another current application that is being looked at more seriously. Some patients have already undergone DBS, but it's actually in clinical trials to use it more often in PTSD. And in trying to relieve this, most people would consider this to be ethically responsible to relieve the pain and suffering. But outside of that normal range, as I mentioned, the possibility of enhancing happiness confronts ideas about what it means to be truthful to ourselves. In a similar way, I want to look at the issue of manipulating memory by electrical stimulation. And memory is an interesting process. Since historical times, we've been trying to figure out what is the process of memory. How does it work? Why does it sometimes fail? Some would say that our memory is what makes us truly human. It gives us the capacity to act upon what's happened in the past, to predict the future. It allows us the capacity to have intellectual thinking, to form associations with other people. And we have learned a lot about the neuroscience of memory in the past 50 years. We've made some critical advances, and I would say in the past 10 years, there have been absolutely amazing advances in our understanding of learning and memory. And they involve the same principles that I've been talking about, the idea of functional organization of the brain, knowing where the memory is being localized and being encoded, and it also involves electrical stimulation. So I want to introduce 3 studies that are along the same lines that tell us something about how neuroscientists have manipulated memory in different brain locations using electrical stimulation. And notably, these studies do advance our understanding of the biology of memory, but they also present a scenario where we might look at whether the technology could be taken from the lab where it has been used on rodents and whether it could be applied to humans. So let's take a look. To talk about this, I think if I just turn these lights up just for a moment. So I want to describe how these experiments were done. All of the experiments have this similar process where adult mice are placed in a novel context. Sorry, we've got a little flickering of the light. It's probably bothering people. Well, that's not good. This didn't happen before, but okay. Better? Okay. So adult mice were placed in a novel context. I'm sorry about that. With unique odors, colors, textures, objects. And mice, like all rodents, are naturally very exploratory. When placed in this box, they explore the environment, sniffing around, touching things, moving around. However, if the context of this environment is such that they are delivered a mild foot shock on a grid that is placed below the box, then they will remember that that box is a box that I received this foot shock, and they will be fearful of being in that box. And if they are removed from the box and then replaced back into that box later on, they will go into a freezing behavior. So rather than being exploratory, they will stop their motion and freeze, okay? So this learning is referred to as contextual fear conditioning. It's a type of classical conditioning. They're being conditioned to the context of the box associated, where they had this associated foot shock at one point in their life. Rodents demonstrate this behavior of learning in this environment to a high level, so it's something that is easily studied in the lab, and the general regions of the brain that are involved in this behavior are well known. So in recent research, and I'm only going to talk about 3 different studies, but they're all along the same lines, and they asked whether you could change the electrical activity in certain brain regions and therefore change memory. And then I'll go back to this so we get rid of that flickering. So one of the questions that was asked was, can a memory be reactivated? And here's our mouse, and is wondering, can the memory be reactivated? A memory that they had before. So this is work done by Steve Ramirez and Xu Liu at MIT. They're now at different locations, but they did this work, and it's incredible. I do have to say that in all of the experiments I'll talk about, they're making use also of another technology, not applied to humans yet, and that is that the mice were encoded with a genetic reporter, something that could be activated when cells underwent strong electrical activity, as they might do when exploring a particular environment and developing a memory of that environment. That's actually a known property of cells, but if it doesn't work, then this whole process doesn't work, and it does. So it actually does help that there was a lot of prior research that went into this assumption here. So as the mouse explored the environment, the brain cells that became activated during the memory formation could be identified by the researchers because of this tag, okay? The mouse was allowed to explore box A and received a foot shock, which generated a memory of the context of that box, the fear conditioning. That fearful memory would typically lead to freezing when the animal was replaced in box A. But because the cells that were associated with forming that memory had been labeled, the scientists could actually stimulate that cells outside of that context electrically. So they take the animal outside of the context, they electrically stimulate the cells that they knew were involved in developing a memory of box A, and when they electrically stimulate those cells, the animal freezes. So what they showed was that they reactivated a memory that was established in the brain. This is actually really important. It shows that memory has actually a consolidation phase and a phase of retrieval. And the memory had been consolidated, but they found a way to retrieve it through electrical stimulation. Okay, so that's one key experiment. The next one, this one received a ton of press last year when it came out. Headlines even in the New York Times about scientists create fake memories. You may have seen some of this. The question that was asked was, if we can manipulate memory to reactivate it, can we also create a false memory in an animal? And the same researchers worked on this using the same general methodology that I just explained. The mouse explored a novel environment, box A, and formed a memory of it. That box was not associated with a foot shock. The animal was then moved to box B. While it was exploring box B, the researchers were able to identify the nerve cells that were activated in box A in exploring the environment because they had this tag on them. So they could activate electrically the nerve cells that are involved in creating the memory of box A, and they did that at the same time that they delivered a shock in box B. Now, of course, the animal had fear conditioning to box B because that is where they received the shock. But interestingly enough, when placed in box A, the animal also had the freezing behavior. Presumably, if the animal is taken to a novel box C, that is a benign box, that it does not have freezing behavior. But what they have shown here is that by, by, uh, by encoding a memory of shock in box B at the same time as activating electrically the cells that told the animal that it was in box A and exploring box A, that when they put it back in box A, they had now transferred to it this false memory that box A is one that I should be fearful of. That is the creation of a false memory. And finally, I will tell you about one other one, and this is by a different lab. This is from the lab of Jocelyn and Franklin at the University of Toronto. And this one, mice were genetically manipulated so that the cells in a particular brain region would not be functional if they became highly active. And again, cells will become highly active during the exploratory phase of exploring the context of the box, as might occur during the formation of a memory. So mice were put in box A and were trained to learn that it was associated with a foot shock, and they developed the condition fear to box A. And then, after forming that memory, these overactivated cells that were involved in forming the memory became inactive because of the genetic manipulation that they had. Once those cells became inactive, if they then put the animal back in box A, they find the animal doesn't freeze. And it should have remembered that that was a foot shock, but remember, the cells that were involved in forming the memory were made inactive electrically. And so the memory had been erased. So what kinds of questions might this raise for us? If memories can be enhanced or erased, how do we decide which memories should undergo this and when to apply these sorts of manipulations if we were to think about applying this to a human condition, which is not directly possible right now. Memory is just one component of our identity, but we tend to think it's a really important one. There's perhaps no more obvious example of this, but when we start to see individuals who lose their memory during something like Alzheimer's disease. This loss creates a loss of awareness of who that person is in relation to others and to the world. Once that awareness is stripped away, then we experience a loss of the person, even if the human is still present in the body. I remember when a grandmother was declining due to Alzheimer's disease, and a family member remarked, "You know, that's not really grandma. It's just her body, but it's not really her anymore." And I think what that summarized was that even though she still existed as a human, she had lost her identity to herself and to others. We should consider, though, if neurotechnology could be used to restore identity by restoring or reactivating memories in a directed way, similar to the technology that has been used in the mouse, with some changes, of course, but on the same principles. It's not likely to be practical, actually, because in Alzheimer's disease and other forms of neurodegenerative disease, one of the reasons for the memory loss is that the cells that are involved in either encoding or retrieving the memory are dying, and dead cells can't be activated. You could put as much electrical stimulation as you want. If there's no cells to be activated, it's not going to work. So there are certainly some hurdles to overcome, but it would have to be a different method than was used in the mouse experiment. But conceptually, if we're interested in restoring memory by electrical stimulation, it may be possible someday, and we'd have to consider whether it had the consequences that we wanted or perhaps unintended consequences. Could it be that we restore some memory and we restore some identity, but it's not the same identity as the person had before? What would we say to that? Would we say that we had created a new person? Electrical stimulation can be used to erase memories, as shown in the mouse experiments. This is actually something that is considered to be more doable in humans right now and is being explored for the possibility of use in something like post-traumatic stress disorder, severe trauma in which the person has developed a memory of experiences that are so severe that is completely debilitating to their lives. Clearly, there's a compassionate argument to be made here that dampening or erasing memories associated with severe trauma should be something that we should consider. However, we also have to consider that to be human is to experience a range of emotions, and some are severe, but under what conditions should we allow the dampening of memory or the erasure of memory to occur? Does any of this run the risk that we would obliterate memories that would allow us to, that would force us to say that we have destroyed what is true and what has really happened. If an event can no longer be recalled, we might actually ask the question about whether it actually had happened. And there's always the risk for manipulation for undesirable purposes. We should keep that in mind. Any technology that can be used in a humanitarian way could also be used in a way that negates personal autonomy. Could memories be inserted without our permission? How can we ensure that technology improves the clinical symptoms of a debilitating disorder without introducing methods that could be considered to be mind control? In the end, we don't have clear answers to any of these things. But I don't think we know what is the right thing to do. At least I don't. But I know the first right thing to do is to ask the questions, because typically what happens is medical technology proceeds much faster than a consideration of the ethics behind that. As an example of this, in searching the literature for scientific articles on deep brain stimulation, what you see is that in the past 17 years, a marked increase in the number of scientific articles that have been published. But if you look to see which of those address ethics, it's pretty poor, okay? And a further example of that comes from a different technology that I'm not talking about today, but just to show you how this can really skyrocket, is CRISPR gene editing. So I did the same thing for CRISPR gene editing, which, as you know, is a technology that when it was developed first, people did start to raise ethical concerns, but there's not really been any ethical decision-making associated with it. And now, in the past year, we have heard of the development of two babies in China who have undergone editing of their genome using CRISPR technology. And again, the relationship between the number of papers published in scientific articles and the number that are actually addressing ethics is really low. So while we don't have answers to all of these things, I think it would be better to have discussions about neurostimulation and its impact on the human condition before the technology is used to erase memory and insert false memories in humans. So I'm hoping that stimulates some ideas for discussion, and I'm going to stop there and thank you for your attention. We do have two microphones in the audience. Ryan has one, and Dr. Brian has one, and we can circulate those if people want to have a discussion or ask questions. We'll do our best. If you could announce who you are, that would be great, because then I could also learn.
My name is Phil Chan.
Okay.
I'm an artist, so just give you a little bit of background. Yes, this does concern me because I was going to ask a slightly more convoluted, complex question, but I'll try to cut part of it out. And it seems to me exploration is an important part of existence, right? Mm-hmm. In animals, it's a relationship between the animal and the outside world, primarily. But in human beings, we also have ideas, which is exploration of interior ideas with each other. Mm-hmm. It seems to me when you're talking about one of these what were these nerve cells? Mm-hmm. You know, there are connectors, and the connectors pass through chemical exchanges, right? And it seems to me one of the reasons why it has these connections rather than just a long rod, okay, just one of the reasons, I'm sure there's many others, is that in the pass-through, it's actually filtering out things so that certain information is passed through in the presence, and other information is not passed through and is absent. So the relationship between presence and absence is a very important part mm-hmm. of this thing, right? And it seems to me that is how you filter, in a sense, ideas that pass through or don't pass through, and through several of these layers, you filter out information, just like like breaking a code or whatever. If what you've done is you did with these mice, this sort of like freezing and whatever, then the relationship between the absence and the presence, it's a very important procedure, and I'm not sure that we understand that when we have deep brain stimulation or whatever it is, that we haven't freeze the situation as it is, and therefore that we're structuring the pass-through, the relationship between the presence and absence in a particular way that is now. But one of the fundamental part of existence, whether it's an animal or whatever, is the constant changing and exploring. And if you freeze that in a way, then in a sense, you're frozen the presence, and you're in a sense not exploring anymore, at least in terms of ideas. So I'm wondering, like, does anybody know about the side effects and how the balance between what passes through and what doesn't pass through and how it evolves is an important aspect of development and existence?
Well, you're bringing up some important concepts. One of them is that the complexity of the electrical activity is, of course, greater than what I use today in my demonstrations, right? So there is filtering that occurs. There's also signals that get stopped electrically by the nervous system and chemically. So not everything that gets started in one place ends up as a signal that another part of the brain receives. So this does happen. There's a lot of modification of those signals as time goes on, right? So that is one concept I think that you were implying there, that there's a greater complexity to this, and that's understood. One thing I will say about the freezing is a behavioral manifestation of the animal experiencing a recall of the memory that I didn't like what I got in this environment, I'm afraid of it, and I'm afraid to start moving around because there might be a sense that it will happen again, or my movement induces this. Who knows what the animal is thinking? But it is just their natural behavior, even in the wild, that if they have, you've seen a lot of animals do this. I see rabbits do this a lot, that, you know, if you see them in your neighborhood or something and you go up to them, they actually freeze. They're perhaps trying to make sure that you don't see them, that the movement will stimulate a predator, perhaps. So these are survival instincts, probably, survival that is present in all animals. So the nature of this is that you know, there are a lot of changes going on in the brain during memory, and it's not to say that we know all of them. What we know is cells that are involved in encoding the memory. We know cells that are involved in reactivating the memory or recalling the memory. We know where they are. We know what groups of cells they are. They can be labeled, and they can be re-stimulated or inactivated such that certain survival-related memories, which are perhaps among our least complicated sorts of memories, the ones that are almost like reflex behavior in a way, that these are the ones that can be modified readily. The more complex ones haven't been tried yet. Animals that have to do a multi-part task and learn to do multiple things in a row, or something like that, hasn't been tried. So by simplifying the system, by using an animal that shows a simple behavior and it is readily trained to do this, simplifying the system, you can get at some of the basic principles underlying these physiological changes that are occurring. You would have to scale that up immensely to talk about more complicated behaviors in a more complicated and brain such as a human brain. Does that get at some of what you were talking about, maybe? Okay. Well, there was a lot in there, so we can talk some more later, too. Yes? Okay.
I've always been interested by the argument that if perhaps a profoundly terrible experience can forever change a person, then also perhaps a profoundly positive experience could change a person. Speaking most specifically to PTSD. And I noticed a lot of your discussion was focused more on physical, mechanical interventions of electrodes.
Yes.
But I was wondering how familiar you were with the literature associating perhaps MDMA with PTSD or ketamine metabolites for the treatment of depression, And I was wondering if you think that might be some snake oil, if you think that there might be some promise there, and if there's anything within the greater community that either needs to shift or even needs to stay the same.
Right, right. So those are good questions. So what you're asking really is about, when I talked about these two neurons communicating each other, in a very simplified diagram that represents what happens throughout the brain. We have electrical, chemical, electrical. Because I study the electrical activity of the brain, and that's something that I can speak to from the research perspective, I wanted to really focus on that electrical aspect. And it's also the newer material, and it's also the one that gives us a lot of precision because we can place electrodes in specific areas. It gives us the ability to turn it on and off regularly just by turning the dial, the switch, and also to control the amplitude of it. So there's a lot of interest in using manipulation of electrical activity. But what you're also referring to is the place where the electrical turns to chemical and then back to electrical. That chemical part of this interaction is certainly a place to also manipulate the activity of the nervous system. And that's been done for many, many more years. And some of it's somewhat successful, right? Some patients get a lot of relief from certain drug treatments. But it's not going to be as precise. You can't deliver it directly to the brain area that needs it. Other brain areas are going to get it too. You can't turn it on and off as easily because the body is going to metabolize this, maybe change this chemical into something else, and its lifetime of existing as the chemical that you want it to exist at is going to be shorter. So there's going to be some challenges there, right? And there are. But that is the first line of attack for virtually all of these. I won't speak to the specific drugs that you've named because I'm not as up on that literature, but I know those drugs, and yes, they are useful. But what people find is that over time, they're not as useful as they might have been in the beginning, and for some patients, they're not useful at all. And so we do need alternatives. And sometimes the electrical activity can be one of them. But even that doesn't work for everyone either. Does that address your question? Okay, good. Glad you're up on that information.
I appreciate your scientific work to help people who are suffering. And as a scientist, you come here, talk to us, a lot of humanities people, a liberal arts school. Why are scientists so deficient? You showed in your graphs that they have no common sense of what the possible dangers are. How can you get your colleagues to have some more ethical and moral consideration? Should they all take humanities classes?
They absolutely should do that. They absolutely should take humanities classes. Thank you for the question. One of the things I do personally is that even in my very first semester of freshman science classes that I teach, I try to introduce considerations of ethics, not just research ethics, because they are learning to do research, and that's a component of ethics too. How do we do research in an ethically responsible way? But also, if we're doing research that has any applications, how do we think of those applications in terms of their application to humans? So I try to get the students thinking about that right away, and I do find that sometimes that spurs them to go on and take other courses in ethics and philosophy and humanities courses. So that's helpful. I'm trying to do my part. I think that I don't know exactly what the medical school training is right now and how much ethics they get, but I hope they get some. I hope they get a lot. I do think that as scientists, it's sort of underplayed a little bit for sure. And people are not necessarily looking at these things as they're going to be the ones to directly apply it to humans. They're doing the basic work. Ramirez and Liu, they're doing work to figure out how does this all work? How can we modify it? Is it modifiable? What are the conditions under which it's modifiable? This tells us a lot about the biology of how the system works, and that's what they're going after. They're not going to be the ones necessarily to apply it to the human condition. I will say, though, that if you go to Steve Ramirez's webpage, it's very interesting that he has a comment on there on his webpage. His little parting comment for people looking at all of his material is, it's not if we can apply this to the brain, it's when, okay? And he doesn't say what species he's talking about there, but the implication, I think, is that what they're doing is something that they do expect someday we can apply some of this to the human brain. It's not if, but when. And so as in the ethics world, we would have to say, well, and should we? So.
Well, I have background in medical dental, and we do get tricked
Okay,
in ethics.
good.
And we're told not to do any harm.
Okay,
I think it's the scientists, the research scientists, that are pushing the envelope, and without any regard of the possible fallout. There's tremendous fallout we're facing. Science is out of control. Who's overseeing what you're doing? Where's the moral compass? Dramatic changes have to occur, not on the level of a professor teaching, but the deans and the people running these schools. Look at the horrors that are going on. Look at what's happening here in San Diego at the VA hospital. All this horrible stuff going on in the news. Look what China's doing with CRISPR technology. This thing, the Pandora's box, has been opened to such an extent that it's we're really challenging our humanity and our survivability. Something has to be done. I think it's the responsibility of research scientists to clean up their own shop.
Yeah.
They should be asking for oversight.
Yeah.
It has to be much stronger. There has to be bite
Well,
to it.
I appreciate your views on that. I would say that on a perhaps a less extreme view, I share some of that, and that's why I'm interested in exploring this too, and why I'm concerned about graphs where we see, the science papers without the ethics. Why aren't we talking about these together? But I do think that we're also going to learn some interesting things about how our bodies work, and we're going to continue to explore science in ways that that increase our understanding of our own human bodies and other animals and plants and everything else that is studied. So I think that's all important, but I do think we have to think about the applications, and that's why you have to have science and the humanities, right? There were some other hands up. Yes.
I'm a law student here.
Oh, good.
In cognitive science.
Okay.
And I was just wondering, I feel like we kind of know it's true that the brain naturally sometimes creates false memories that we think are true, and then they turn out not to be true. And even our normal experience of memory are kind of subjective, they're based on contextual factors, your emotional state at the time, your personality, and every time you think about that memory again, it also is affected by those things when it's re-encoded. So
Yeah.
I was just wondering if you think that fact is a good argument for scientifically manipulating someone's memory to be ethical, because our memory in the first place isn't an objective depiction of reality.
So it's already being modified, so if we modify it some more, how could that be a problem? I thought you were going to go with a different angle, whether this had any relationship to whether we could trust our memory when we give depositions in court and things like that. So I think what you're pointing out, though, is the fact that memory is labile. I mean, the biology shows us that, how easy it is through electrical stimulation or chemical, if we go to chemical techniques, which have certainly been used as well. I mean, patients who experience PTSD, if given within the first 6 hours a drug called propranolol, which is a beta adrenergic blocker, will have a depressed memory of that particular traumatic situation. It's not always possible to do that. You don't have the drug available 6 hours. You didn't know when you're going to receive this traumatic experience. But how does that work? It's depressing the sympathetic nervous system, because if you activate the sympathetic nervous system at the same time that you're experiencing an event, the memory is a little stronger. You know this is true. Things that you have experienced that were very emotional are things that stick with you longer. So memory is labile, and the context does matter, and the biology shows that it doesn't take that much to shift it one way or the other, to erase it, to create it. And so I think that it has implications for criminal law, and I suppose it has implications for your argument, such as it's already happening naturally, so how is this even changing anything if we just go in and do it? But I think what's happening there is that you're imposing a human decision then on that change, and you're making a value judgment about what that change is going to be and what you hope the predicted outcome will be. Whereas when that happens naturally, yes, it changes, but you're not imposing a human will on that. So perhaps distinctive that way. Let me come back to you. I thought there was another couple back here. I want to make sure I get, yes?
I have maybe two quick questions. I'll try to be really quick. In the example where you talk about reactivating the memory,
Right.
Is that really reactivation of a memory, or are you just stimulating the location in the brain that you know is linked to a memory?
Yeah.
Sorry,
Yeah, no, that's a really good question. It's a reactivation of the cells that were somehow involved in processing the memory, and then that somehow produces the behavioral manifestation of the animal remembering that environment. You could argue, like you're saying, that it may not really be a reactivation, but we're just activating those cells again. The idea that they're promoting through that research is a concept that I think is subject to debate in neuroscience and psychology, but it's the concept that every time you recall a memory, you go through the process of a process by which the memory is labile for a period of time, and it's sort of reconsolidated. And that process involves some of the same circuitry as was involved in processing the memory to start with. So there's something about that, I think, in that literature.
So my other question, this is more relevant to my interests. I'm a communication scholar.
Okay.
I do research about how people communicate about dying and death, and
It's a good question. So how do we define what is natural or normal? In terms of clinical applications of technology, whether it be pharmaceuticals or deep brain stimulation, it is defined by the interaction and the communication of the patient and the physician to define what is normal and to look at whether that person has, sort of a neutral affect, one that is slightly sad or slightly happy, or some extremes of that. And where is the dividing line of that is up to the patient and the individual. There isn't any other way to do it. It's often based on the patient's self-reported affect, on scales. On a scale of 1 to 10, how happy are you today?
There's research and scholarship that drives a lot of what, this is the biomedical kind of understanding of disease and illness processes, and whether or not grief is a normal response to death.
Yeah.
People would say it is normal,
Right.
and that there's some other outside force that's saying, well, you shouldn't perhaps be this sad. I think there are implications for how we might redefine, right? If you can eliminate, as we're describing, someone's grief experience or their trauma experience, then you are redefining what is traumatic or what is grief or what can be grief.
Right.
I think that's pretty.
Yeah, that's some interesting points. Yeah, I'll have to think about that one a little bit more. But yeah, that's really interesting. Yeah. Yes.
The case that you mentioned about the 2 babies in China
Yes.
that were genetically modified, do you know if it was for aesthetic purposes or for preventative of a disease? And then do you think that line is important or like a compromise could be driven?
To my knowledge, that was for a single mutation that was a disease encoding. That's my understanding of it. Probably should know a little bit more about that particular literature, but it hasn't been published or anything, so there's very little information available. Do I think that there's a dividing line between things that are changing something for disease versus changing something for aesthetic reasons? Yes, I think there is. I think that we do use other mechanisms to modify someone's symptoms or chances of developing symptoms. And gene editing is one that can do that. I'm not really sure how I fall on that right now, but one thing about gene editing that we always have to consider, which is different from using any other form of therapy that we were talking about, either medical, pharmaceutics, or electrical stimulation or anything like that, is it's not just changing that individual, but it's now changing the genome of that individual in a way that can be passed on to their progeny. And so you end up with this sort of the domino effect, right? How can it be an autonomous decision for the children of that person to decide if they want gene editing when, well, these were embryos that were decided. They didn't get to decide either, but I think you see the point, that there's a downstream effect of this that is different from treating one patient at a time. And so that's one of the big challenges with gene editing. And of course, there's also the challenge that you're doing it to an embryo. So.
But the same could be applied for the neuro or electro treatments, like for happiness versus Parkinson's disease.
Oh, in terms of, yeah, whether it's a clinically, deemed to be clinically necessary to allow the person to live without those symptoms versus someone who simply wants to be happy all the time. There have been cases where people who have been treated for something like Parkinson's or OCD or other disorders that they asked physicians apparently to try to manipulate their happiness, that they knew that they could potentially work that in somehow. Can you just make me a little happier? I'd like to be happy all the time. So, yeah, physicians didn't feel that, the physicians didn't do it, from my understanding, but there's a risk. Yes. Oh, sorry. Did you have one also?
One, have you seen the film Eternal Sunshine of the Spotless
So
Mind?
I haven't seen it, but I know what it's about. Yes, I guess I need to see it.
I used it with my students in
Yes.
a sexual ethics class to talk
Okay.
about, this question of if you could
Erase memory.
erase the memory of a bad breakup.
Yeah.
And it's very provocative. I
Yes.
think you would find it.
Yes. I have a long list of things that I need to read or view.
Another question I had is, do you sense alleviating suffering. I'm deeply concerned that there's creeping ableism and an inability to recognize the dignity of those who are disabled.
Yeah.
And to recognize the value that they bring to our communities. And I wonder if the lack of ethics assessment in some of the journals is in some ways, just an example of this kind of creeping ableism. And if that's so, if there's something there, what do we do about it? Or where do you see the conversation going so that we could perhaps push back on that and recognize the real value of our disabled community members and those who are most vulnerable?
Yeah. No, I think you bring up a good point, and it's kind of in a similar line of, what is the normal amount of grief, and what we, and how does that impact, our relationships with each other even. When you talk about the abled and the disabled, having a diversity in the community, how does that expand us in terms of people? I mean, it expands us for compassion and dignity and empathy and other sorts of things that we value, right? But without that, would we just be these, the robots of Gattaca or something, where we're all, you have the haves and the have-nots, and it's challenging. I don't have the answers to that. I mean, I think this is something that we have to struggle with, but it's definitely a really important topic. Thank you for bringing that up. Did you have one also?
Yeah, I'd have a lot to say, but
Okay.
I'll try to keep it brief. So
Okay.
I'm Sonia, and I'm just a member of the general public.
Okay.
It's very fascinating what you're saying. Thank you.
Thank you for coming.
So you've addressed the introducing electricity to the brain.
Yes.
Talked a little bit about the chemicals.
Yes.
But what I've seen in my experience, and there's clinical studies on this, for example, hypnosis. And that in terms of changing behavior, taking away memories, I don't know about anybody memories, but taking away memories, changing behavior, canceling things out, to the point of being able to perform surgery on somebody while they're under hypnosis, they don't feel pain.
Right.
So I don't know where that falls, but that's a whole other thing to address, which would be interesting.
Sure.
And then the other thing I had to
Well, it
say,
definitely falls in the area of mind control, right? Hypnosis. So.
very powerful. And then you get this whole thing of, well, when you're raising a child, it's all mind control because you're telling a child this is et cetera, et cetera. So,
Oh, is that what it is?
I mean, what you're teaching people as values, I don't know where that line is, but that's a whole other discussion. But I would like to hear what you have to say about hypnosis, for example, and that has been clinically studied.
Yeah. I wish I knew a little bit more about it. I will tell you that we were actually just talking about hypnosis today because I had brought up the fact that a colleague of mine who is a physician revealed to me that in medical school, I don't know about your medical school training, you're a dentist or a?
The first 2 years, medical and dental was all about.
Okay, so he revealed to me that his medical school training at Harvard included training on hypnosis, and that physicians were trained in the process of hypnosis so that they could use it on patients. Excellent, but well, I'm glad that if they're doing it, they're trained at least. But the point is that when would they use that? Would they use that because a patient was, as you say, experiencing pain, and you could use it to reduce the pain? But could it be used to manipulate the decision-making capacity of the person, such that, well, they really don't want to have this technique done, but I think it's for their benefit. So if I hypnotize them and talk them into it, maybe they'll do it. But now that mind control is removing autonomous decision-making, and we typically value that process. So I don't know how I feel about hypnosis. I know that there are people who have some capacity to do it, and I know there are some people who are more easily hypnotized than others. And if you are one of those, don't ever volunteer to get up on stage, or you're going to end up, lapping up water like a dog or something. But I think that there is a capacity to do that, showing that, if people can experience that, then it means that the neural circuitry can be modified simply by suggestive language of others, the communication. How is this even possible? It's amazing to me that it works.
Because of the reception we have outside, we've got time for one more question.
Okay.
And then we can continue this over a drink
I'm
just
so
outside, to
excited.
which you're all
I'm so excited.
invited, of
There's
course.
so many questions. I'm going to get one from someone who didn't get a chance to ask yet.
I have two small ones.
Okay.
If that's all right. First, going off of the hypnosis portion.
Okay.
I was wondering what your thoughts are on suggested memories in hypnosis. Like, I've been studying some sort of portion of suggested memories
Okay.
And so what your thoughts are on the ethics of using some of these forms of therapy
Hmm.
to possibly erase memories
Right.
implanted by therapists
Right.
dissociative identity disorder. And then also my other question was your thoughts on this possibly becoming more of a quick fix solution for some people who obviously aren't suffering from some sort of debilitating magnitude of these illnesses, and then
Right.
doing it in that area, because we've seen in some people, specifically with PTSD, that some people can encounter severely traumatic events and process them in a way that allows them to function normally, and others to develop PTSD.
Right.
And so what are your thoughts on that ethically?
Yeah. So now we're also straying into an area that sounds very clinical to me, and I'm not a clinician, as I said. But I think that in terms of the ethics, I think it's something we have to look at extremely carefully, because we know that the brain is labile. We know that these memories can be manipulated, and who's to say that one individual should have the opportunity to do that? Obviously, they're doing this because they think it's for the benefit of the patient, but why do they get to make that decision? Who decides this? And under what conditions would we allow this sort of thing to occur? I know it does occur, and I know that there are going to be some patients who are more susceptible to it, because there's also patients or people who are more easily hypnotized than others. And we know that the brain can undergo these changes. And so it's possible that through suggestive insertion of memories that you could create a false memory in a patient, that you could create something that they think is a memory, but it's not really a memory. So again, the issue of truth, right? This makes me think of parenting, actually, because I still remember cases with my own daughters where most people don't have a memory of anything that happened before 3 years old. In fact, many people, it's like 6 years old. But what I find is that my daughters think they have memories of things that happened when they were younger than that. And the reason is because they've been told stories. It's been suggested to them. They've seen pictures about what happened in situations where they have no actual memory of it, no true memory, but they have created a story that sounds like a memory to them. And that's kind of interesting to me, because I think that falls along those lines, right? So some great ideas. I'm so glad that you all came and really made me think hard about this too. Thank you so much.